morning. I'm Ilse Kuffner. I'm a PI over at the USGS, and I'm going to introduce Nancy Prouty, who is also with the USGS at our Pacific Coastal and Marine Geology office in Santa Cruz. And she has been with the USGS since 2007. She started out as a Mendenhall, and then in 2009 converted to a PI. And her work focuses on paleo recorders of land use change and climate change, and she focuses in two areas, in shallow corals and in deep sea corals. So I think she's going to talk about her deep sea stuff today. Her education, she did a master's degree at the University of New Hampshire, and then her PhD was at Stanford with Rob Dunbar. And let's see, what else did I want to say? Oh, there is a TGIF after that was thing I was told. Yes. So,
well, after
thanks.
Rob Dunbar, that's what they say.
All right. Well, thank you very much, and go for it.
Thank you. Thanks, Ilse. I was going to start off and apologize that I was sitting between you and your beer, but I now realize that you drink during the seminar, so I don't need to apologize. So,
we'll never have it. We can have one.
Oh, that might help. Thank you for the invitation to come and speak at your joint seminar series. It's been a privilege to speak with both USGS and USF folks over the last day and a half. And I, after meeting with people, I have to admit I feel like a bit of an imposter speaking to you about the Gulf of Mexico, where I'm sitting, in my Santa Cruz office, and many of you have been doing Gulf research for many decades. So, with that disclaimer, what I hope to do today is to give you a story about the Gulf of Mexico and the Mississippi River, but from a really unique perspective, and in this case, it's the deep sea corals. Two people that I want to just give a thanks to and acknowledgement are USGS personnel, Noreen Bester and Chuck Holmes. They really planted the seeds for this initial work that allowed me to grow with it. So I really thank them for being generous and sharing samples and many radiocarbon dates as well. So this study that I'll be presenting today was published in January in Global Biogeochemical Cycles. And let's see here. It's really a culmination and contribution from many co-authors from a variety of institutions. So I just want to acknowledge them. Brendan Rourke, who maybe some of you know at Texas A&M, Alan Koenig at our USGS Denver Center, Amanda Demopoulos, who's our project chief for the deep sea coral work, two USC folks, Fabian Batista and Matt McCarthy, Ben Kokar at Stanford Synchrotron, Dave Selby at University of Durham, and Furu at NEOS. And again, our funding agencies, of course, USGS, collaboration with BOEM and NOAA as well. So as you know, the Gulf of Mexico, Mississippi region is a really unique place to work, particularly because there have been so many changes in the region. And for me, this backdrop of research is a really nice setting for me to start asking new and different questions about the region. So the area has been termed the America's breadbasket, and this is really driven by the fact that 65% of the land is farmland and 25% of it is harvestable cropland. And it occupies 40% of the lower 48 states. And it's the third largest river basin in the world. It contributes 80 to 90% of freshwater into the Gulf of Mexico and contributes about 30% of the nitrogen loading to the North Atlantic, with about 50% of that being attributed to nitrate. So just to give you a sense of comparison, today's nutrient loading or nitrate loading is about 5 to 6 fold greater than natural background. So there's a real legacy of nutrient loading and land use change in this region, and this is really related to land disturbance. So within the Mississippi River Basin, you've got land clearing due to agro-industrialization, and this is related to folks settling down and putting down crops, draining the land for farms, and this is intimately linked to the amount of sedimentation coming out of the river and eventually being deposited into the Gulf. So there's a large amount of soil erosion, and linked to that is the installation of the dams that seem to have reduced the amount of soil erosion. And of course, this land disturbance and land use history is intimately linked to the nitrogen cycle in the region. So over the course of history, for this period, you get a lot of nitrogen fertilizer use, and this is related to release of stored nitrogen and then obviously synthetic fertilizer. So within this region, there's a real history of human activity related to both land disturbance and nutrient loading. And this really mirrors the global cycle of nitrogen, where there's a greater flux of nitrogen as a function of manure production, fertilizer production, and also crop nitrogen fixation. So the common link here between nutrient loading and land use change really is the persistent ecological problems that the Gulf of Mexico is experiencing. And as many of you know, this manifests itself into hypoxia or anoxia, and in extreme cases, of course, the dead zone. And I understand now that the dead zone has reached to a size of the state of Massachusetts. Obviously, it's the loading of nutrients followed by eutrophication and then a kind of a collapse of the food web. I also understand that the task force in the region is really aiming to reduce this size from over 22,000 square kilometers to something closer to 5,000 kilometers, and perhaps doing this by change in fertilizer use, a reduction in nitrate transport, and maybe through modifying agricultural practices. And this could be done by changing the types of crops that are used. For example, in this case, corn and soybeans contribute 52% of the nitrogen loading. So there's a real human signal in the nutrient loading. But what's interesting is that this isn't unique. These hypoxic periods are not unique to the present day. And folks at the USGS, including Lisa Osterman here, has done work showing that these hypoxic events occur in the historic past. This is work by Peter Swarzinski. I believe Lisa was also co-author on this paper, where they were able to document hypoxic events using redox-sensitive elements. So hypoxia is a natural phenomenon, and they can trace it back in sediment cores using these redox-sensitive elements. And then Lisa's done work looking at these low oxygen-loving forams and seeing that their distributions have occurred in the past, but that they are an accelerated occurrence in the last, 100 years. So there are past hypoxic events, and we can see them in the sediment records. So the question is, are there other archives out there that can also be really well dated and that can help answer maybe response times to these changes in water conditions that are related to land-based activities? And so what I hope you walk away with today is that a real promising archive, a very sensitive one, are deep-sea corals. So I imagine many of you here study shallow corals, and you're familiar with calcium carbonate corals, like Lophelia here. They build their skeleton out of calcium carbonate. But there's also a whole group of deep-sea corals that do not build their skeleton out of calcium carbonate, but instead build it out of proteinaceous material, so organic material. And if you're interested in more about sort of the utility of deep-sea corals, we published a paper last year in Deep Sea Research outlining sort of the spectrum of types of corals and some of the proxies that we're using in these deep-sea corals. So the focus of my work is the black corals, the proteinaceous black corals, the Antipatherian. And I don't know, I think, Chris, were you on the cruise when that one was collected? Okay. Let me just give you a little bit of overview on these black corals, because I think they're fascinating and they really hold promise to application in the paleo world. They're very much more like a tree. They look like a bush. They display radial growth such that the portion of the coral is in the middle of that. That's a disc, so it displays radial growth. and it has layers. The layers are composed of chitin. It's like an exoskeleton or like your fingernail, and it's interlaid with protein. And you can see in the SEM images and the microprobe that there's real distinct banding to their skeleton. So I think the Ellen Druffel coined deep sea corals as the bristle cone of the sea. And there's great analogies between dendrochronology and deep sea corals. So a lot of work has been done on calcium carbonate bearing deep sea corals, but almost nothing has been done on these proteinaceous corals. So that's where a lot of our work was done to develop techniques to understand how to use these in the paleo world. So the reason I bring up this plot is when deep sea corals were first dredged up through accidental trawling, a lot of them were found growing on orthogenetic carbonates. So people believed that they were feeding on methane-derived carbon because of their occurrence. They need a substrate, and that's actually what the carbonate provides for them, but they do not feed on methane seeps, and that's important. Instead, what we find when we compare their stable carbon isotopes is that these deep sea corals, their delta C13 in both their tissue and their skeleton is more closely aligned with surface particulate organic matter and even algae, in this case sargassum. It's nothing near depleted if they were indeed feeding on methane seeps. So this is important because these corals are feeding on photosynthetically derived material, and this will be a common link that I go back to. And the other reason it's important is that we can radiocarbon date these deep sea corals and not be concerned that they're feeding or that their carbon signal is being disturbed by an old age resuspended carbon. The other important factor is when we subsample these specimens, we can actually capture the bomb carbon in these deep sea corals. So the upper plot on the right, sorry it's a little bit small, is the radiocarbon signature from the flower gardens. This is Amy Wagner's work, and she nicely displays that the shallow coral can capture the bomb C14. If you're not familiar with the bomb C14, basically it's the nuclear bomb testing in the 60s that increased atmospheric C14 substantially. And so the shallow corals are exchanging carbon with the atmosphere very rapidly, and they're capturing that C14 in their signal. But the question is, these deep sea corals at 400 meters, they're not exchanging anything with the atmosphere. So how are these deep sea corals capturing the radiocarbon bomb signal? And if you look at trophic studies, you know that these corals are feeding on a mixture of zooplankton and phytoplankton. So they're about a trophic position of around, between 3 and 4. So they're kind of carnivores. And the zooplankton and phytoplankton are coming from the surface. So basically, these organisms, again, living way below the photic zone, are feeding on rapidly exported organic matter. So for the work we're interested, they actually provide a link between what's happening in the surface water and what's happening in the deep sea. And again, we can robustly age date these. And this really starts to help explain the characteristics of these deep sea corals. So the deep sea corals that we collected were in the DeSoto Canyon. I know several of you are working in that region for your studies, and also the Viasca Knoll region. They were collected about 400 meters water depth. And in a previous paper, we showed through radiocarbon dating that these organisms are extremely long-lived. In one case, we had an organism, a deep sea coral, that was over 2,000 years old. And these are very slow growing. We have growth rates of 8 microns per year. So they display extreme longevities, and they're slow growing. So we would like to know a little bit more about the environment that supports these unique organisms. So we had, with colleagues, deployed benthic landers that were out for a 1-year period. So it really helps us to understand the environment, both in terms of organic matter supply and also flux of material. So basically, it's a sediment trap that's deployed at depth. And you can see in this picture, it's very turbid, and the flux rates here were extremely high. And that begins to make sense. These corals, they need accelerated flow because that's their food delivery mechanism. So we wanted to understand the links between food delivery and just the setting that these corals live in. And I was speaking to Dave Hollander earlier today and just really drove home the fact that these sites are really connected to Mississippi River discharge. You get high riverine input through fine-grained materials that are organic rich in carbon. The upper panel is the pigments versus the fluorescents for our first year of deployment, and you see a really nice coupling between fluorescents and pigments. And so we kind of build this story where you get Mississippi River discharge that's enhanced in nutrients, so there's a strong delivery of organic material. This leads to increased surface productivity, and at depth, then what we see is a response in elevated pigments and fluorescence. And through biomarker analysis, our data suggests that most of the organic matter that the corals are feeding on are probably marine microbial derived. But again, it's all triggered by nutrient loading from the Mississippi River. So this starts to explain maybe why we're seeing that banding in the corals. Again, in the paper that we published in 2011, we counted the bands using the SEM images and compared those dates to the radiocarbon ages, and they were very close. So at this point, when we published this, we hypothesized that the banding in the coral was probably a function of food supply. Shallow corals show really nice banding. I know folks here are working on shallow corals, and you know that's mostly driven by light levels. But again, these are way below the photic zone. So, you know, it's not light levels, but we really do think it's food variability. And when we start to look at changes in food delivery through time, we do see there is interannual variability. So this is pigments and some fluorescence data in the green, and we see interannual variability in the organic matter supply. So we see a change in sterols between year 1 and year 2 and N-alkanes, as well as percent organic carbon. So this is just giving you a snapshot of, yes, there is interannual variability, and maybe the banding in the corals is responding to these changes. We're in the process of looking at maybe how the oil spill impacted food delivery, and that's a whole other study, but I think that's important to understanding how sensitive these corals are to environmental change. So again, this is all geared towards paleo, and we had to figure out how to analyze these samples in an appropriate manner. And they're really conducive to laser ablation because of their size. They're really small, and so we can put them on a petrographic slide, and we can scan them for elemental concentration. So the tricky thing is that the topography of these corals is very complex. So we didn't want to just do one scan for trace metal composition. We wanted to do multiple scans that could account for any kind of topography change. So we ran 20 scans across the coral. And again, this is the oldest portion of the coral and then growing younger outwards. And so what we did is we were able to create concentration maps of their elemental composition. So again, most of the work on deep sea corals has been done with the scleractinians, but this again is proteinaceous, so we had to develop a matrix match standard. And I worked with Alan Cohen on this, and at the time his tech would go out to various places and try to see, you know, what kind of matrix could we use. And so at the time she was pregnant, and she would go to these candy factories because they were actually using similar organic components to make their candy. And so I think she enjoyed that part of it. What we ended up using was a epoxy standard that we added certain known concentrations to, and then we created these concentration maps. And needless to say, the results were really interesting. So this is a blown up version of this concentration map. So this is the outer edge of the coral, and this is, again, the youngest portion of the coral, and then this is 800 microns along the circumference of the deep sea coral. And the fascinating thing was that in all the corals that were collected alive in the Gulf of Mexico, we saw this outer enrichment, so again, the youngest period of this element rhenium. So I was not an expert on rhenium when I started this, and it wasn't the intention, but we just became really fascinated by this enrichment that we consistently saw. We also saw it in the solution ICP-MS, which is just basically discrete sampling. So we were confident that this was not an artifact of the sampling procedure. So I had to do a bit of background reading on rhenium to try to understand, what are the causes of this enrichment and what are the implications of this enrichment. So this is kind of rhenium 101, sort of the what, where, and when. In traditional geology, rhenium is used with osmium for geochronology. It has a natural source, so weathering of organic-rich sediments. And as a result, there's a tight coupling between rhenium and organic carbon. It's, as I showed you before in Peter Swarzinski's work, they've used it in sediment cores as a redox tracer. So it's really an important paleo redox tracer. In oxic waters, you typically get rhenium-7. It's highly soluble and mobile. You see seaweed happens to be really enriched in rhenium, so it bioaccumulates. And interestingly, it has very similar chemical characteristics to technetium-99, which is a byproduct of nuclear fuel processing and, for example, was released during Fukushima. And so many people use it as a chemical analog to try to understand uptake of contaminants. The primary source of seawater rhenium is river rhenium. But what's interesting is that the sources and sinks of rhenium are not equal, such that there's an imbalance in the budget. So there's some idea that maybe we're underestimating riverine contribution of rhenium or estuarine contribution of rhenium. So we basically did an inventory of rhenium concentration in the setting that these corals are growing in. So we measured rhenium in the water. That was from Christian Miller's thesis. We took particulate organic matter sources, matter, and measured those for rhenium, sediment trap, sediment core, brown algae, and then all the way up on the top, this is a log scale, there's our coral rhenium. So the coral rhenium is substantially elevated relative to crustal abundance, and crustal abundance concentrations are pretty close to our sediment, which makes sense. But the black coral is nothing as enriched if it were authenogenic sediments, such from paleo. So the take-home here is that the black corals are really bioaccumulating rhenium. Maybe they're getting it from the seaweed, which has extremely enriched concentration factors, but also